Hello everyone. Today we will be reviewing Chapter 2 and just going over some of the milestones and then also some of the influences that we see on development. And so this chapter is really broken into three parts. The first is genetic influences on development, prenatal development, and then into childbirth. Today I'll be reviewing some highlights of each section, and so really it's more incumbent on you to read the chapter, but also noting some of the highlights that I'm presenting in this particular PowerPoint presentation. And so when discussing genetic influences, there's an intricate balance that we see played out in development between the unfolding of genetic plans and also the environment and its impact on the modification to those genetic plans. And so generally when we're talking about inheritance or genetics, we see alleles that have different variations of specific genes. And this is essentially what makes all of us different from one another. And so when both alleles in the pair of chromosomes are the same, we call them homozygous. When they are different, we call them heterozygous. So when they're heterozygous, the dominant version will actually be expressed, and those are denoted by the capitalization of the letter. Often one allele is dominant, which means that its chemical instructions are followed rather than the instructions of the recessive allele. The recessive allele will be ignored, but the individual will still carry the recessive gene. If they reproduce with another individual who also has a recessive gene and they happen to contribute to it, then the recessive gene will be expressed in their child, as in the case that we see on the PowerPoint slide. So as you can see in the picture, she has a dominant allele that is ultimately influencing their children to have a 50-50 shot of having red hair or dark hair color. And so here we talk about an inheritance process that is specific to influencing the sex of the child. And what we see with sex-linked inheritance is that males are more vulnerable to genetic deficits or defects or mutations, primarily due to the lacking counter gene on Y. So this leads to an increased vulnerability to disease and death throughout the lifespan due to genetic defects such as Fragile X syndrome, where one X chromosome has a defect. So this is more common in males than females because females have this backup X chromosome. So we also see the impact of this sex-linked inheritance on males' behavior, such as risk-taking behaviors and the effects of testosterone on suppressing immune systems. So in addition to genetic influences that we've discussed, the environment also has a significant impact on the expression of those genes. And so genotype is essentially the genetic makeup of cells, and the expression of those cells is the phenotype. So on this slide we talk about the interaction of our genes or genotype in the environment and the resulting phenotype or expression of our genes on the environment. So the example that you see on this slide is actually describing a very simplified process of an individual with inherited traits that predispose them to misuse alcohol. And so this predisposition may include a parent's past misuse of alcohol, prenatal exposure to alcohol wherein the mother used alcohol during her pregnancy, and this having lasting implications on the genetic makeup of this child. So we have this individual who is predisposed, and now we add on the extra layer of them interacting in their own environment as an adult, as a child, throughout their lifespan. So their environment may include parents who continue to use alcohol, maybe as a means of coping with stress or having a true substance abuse problem, all the while this kid is learning that to deal with stress, you drink alcohol to have fun or de-stress. Later, when this kid has grown into an adult and he is stressed with work and family, he starts drinking, as opposed to one who can cope with stress effectively, like working out or hanging out with coworkers instead of drinking. Now, obviously, this isn't the case or is true for everyone that grows up in a family that has parents that may tend to drink more or even have this predisposition because there are many factors that may help to protect the individual from developing a disorder or engaging in any type of maladaptive behavior. So on the one hand, or on the other hand, there are also a lot of risk factors that may impact an individual, such as the example we discussed. So this model is not the end-all, be-all because of those protective factors, such as social support from family and friends that may help to override those genetic influences. So this basic model of input-output is known as the diathesis stress model, that takes into consideration the interaction of genetic influences in the environment on people's behavior. So this is actually used in clinical psychology to conceptualize psychopathology such as mood and anxiety disorders. And essentially what we're seeing is that the environment can turn genes on. And so given that example, you may be asking, how do we actually study the effects of genes? And one particular method described in the in the chapter are ethological studies. And so this method, for example, examines a life history of a species by observing markers or makers of developmental transitions like tin version, or more specifically herring gulls, who produce shiny eggs that attract predators. And as a result of those shiny egg shells that attract predators, they get rid of shells, but not too soon. And so this is one particular example of how ethological studies would examine this developmental transition. So research questions involved in ethological studies may include what makes all members of a species alike, which attributes contribute to common developmental outcomes of that particular species. And so next, the chapter discusses what occurs following conception prenatally within the womb. So the prenatal period is split up into 3 stages or periods involving a germinal state, the embryonic state, and the fetal stage. So the germinal stage of development occurs during the 2 weeks following conception. So it involves the zygote, which determines gender, and each human cell, sperm cell, and each human egg cell contain 23 chromosomes that contain genes. And so the 23rd chromosome is either X for female or Y for male. The male sperm determines the sex of the zygote. So the zygote begins to divide into 2 cells, then 4, then 8, and so on and so forth. And by birth, the zygote has divided into trillions of cells, each containing 23 chromosomes from the egg and 23 from the sperm. The zygote then migrates down the fallopian tube and implants itself into the wall of the uterus. This is a difficult journey where about half of the zygotes don't actually make it, because it is either defective or it implants itself on an inhospitable part of the uterus. During the embryonic stage of development, the zygote becomes an embryo which continues to divide and its cell begin to differentiate. By this point, it's only an inch long, but it has a heartbeat, it has arms and legs. In addition to this, you also can observe the nervous system starting to develop, the digestive and respiratory systems, sensory receptors, skin, and including hair and nails, also begin forming during this particular period. Embryos who have one X and one Y chromosome begin to produce testosterone. The fetal stage of development involves the now fetus developing the ability to move. And also during this time, the fetus increases rapidly in size during the last 3 months of the fetal stage. And while in the womb, the fetus can hear and learn. They can hear their mother's heartbeat, gastrointestinal sounds that are associated with her digestion, and also her voice. And one particular note within here is that each of us are born with 100 billion neurons, and we see that the basic structure of the brain and neurons is formed within the first 2 trimesters. And during the fetal period, the fetus's brain is being further developed by a process called neurogenesis, where it produces new neurons. And connectivity of these neurons develops in the third trimester and continues through years 1 and 2 postnatally. In the womb, androgens and estrogen and also testosterone are being produced. Both males and females have estrogen and testosterone, but just in different quantities. Along those lines, we all begin as female in the fact that without the influence of hormones prenatally, a fetus will develop as female regardless of chromosomes. In order to have sex differences, there needs to be a high level of testosterone in order for the fetus to develop as a male. Given this process, there are multiple steps in this process where things can vary in their development. For example, high levels of androgens predispose the fetus, whether male or female, to develop a sexual preference for women. In this particular slide talks about teratogens that may be or that are hazardous to prenatal development. And so this includes drugs like caffeine, environmental pollutants like radiation or x-rays, toxins that are found in some foods. If mother has nutritional deficits such as iron, this definitely has been demonstrated to impact the cognitive development of the fetus and later the baby or the child. We also prenatally women are needing to take their vitamins such as B vitamins or folic acid. Particular to this, you see that one specific teratogen on here has exclamation points, and that's stress. And so what we see with stress is that cortisol impacts the baby by calibrating the infant's stress response system for the future. So not only is it impacting the infant's stress response system, but also their immune system for their entire lifetime. And so we'll talk about more of the optimal time or the safest time to have a baby in terms of the maternal age. And also one other note within teratogens is considering the mother's predisposition or even diagnosis of specific diseases and how that impacts the child's development prenatally and also throughout their own lifespan. So here we'll move into talking about the actual birthing experience and the different stages of birth and what that looks like throughout the world in comparison to the US. So here from this graph, we actually see that the safest time or age range to have a baby is from 16 to 35. And the most optimal time, the most optimal time given fertility is 22 to 35. So teen mothers tend to have more birth complications and are more likely to deliver prematurely and have low birth weight babies. And you may be asking, why is this? So what we see is that the monarch ovulation gap has yet to fully develop, and we also see an increased likelihood of poor prenatal care, poor nutrition, especially early on when it's the most critical or most important, and we also see higher levels of stress. So we also see that after 35, there is an increased risk of miscarriage that is probably due to chromosomal abnormalities and also a greater risk of birthing complications. And so with this particular slide, we see that birth is a 3-stage process, and the first stage of birth is the longest and is not called labor by accident. So this stage begins as the mother experiences contractions that are typically spaced 10 to 15 minute intervals, and it ends when the cervix is fully dilated so that the fetus's head can pass through. You also see false labor during this particular stage where the mom experiences a few contractions but then they stop. This is essentially a warm-up period to labor. And so this first stage lasts an average of 8 to 14 hours for firstborn children. We also see this transition to it taking about 3 to 8 hours for later born children. And as labor proceeds, the uterus contracts more frequently and more intensely. The second phase of labor begins with the fetus's head placement at the cervical opening. And so the second stage is the actual birth or the delivery of the child. And so the fetus's head passes through the cervix and into the vagina and ends with the baby emerging from the mother's body. So this stage is when mothers are told to push or bear down with the contractions. An epidural or other medication may stunt the mother's ability to feel the contractions, and so they have to sometimes use a monitor to know when to push. A quick delivery may last 30 minutes, whereas a long delivery may last 1.5 hours or more. And so again, delivery time really varies person to person. So the third and last stage is the delivery of the placenta, and this takes about 5 to 10 minutes as the uterus once again contracts and expels the placenta from the mother's body. And so this is done by even more pushing. Once delivered, the newborn is assessed using a quick appraisal called the APGAR. The APGAR is a quick assessment of the newborn's heart rate, their respiration, their color, muscle tone, and reflexes. So each characteristic examined is rated from 0 to 2 and results in a score up to 10. Newborns are also assessed using the Neonatal Behavioral Assessment Scale, which is a test that assesses the newborn's neurological responsiveness to environmental stimuli. So each newborn has 20 inborn reflexes, such as reaction to comforting and other social stimuli that are assessed by this particular assessment scale. So differences you can see across childbirth around the world include settings. Typically, U.S. deliveries may occur in hospitals, at homes, or in birthing centers. In the U.S., we have access to medications that also help to induce contractions, while also we have medications that help to block pain. So support for the mother may actually range in those who are present for the delivery. For example, your best friend, your sister, and or your mother may be in the room, but for some cultures there are cultural practices surrounding pregnancy and delivery. And so this may include the presence or distance of the father and his role within the delivery. And more we are seeing mothers opting for midwives, where this has been a long-standing practice for some cultures in non-Western medicine.